Hello everyone, my name is Arne Schnitt and I'm a PhD student in Germany at the Federal Institute for Risk Assessment. And the topic of my presentation is methicillin-resistant non-aureus Staphylococci in German dairy herds. Background, I think you all heard about methicillin resistance. It is well known from MRSA or methicillin-resistant Staph aureus, which is a major nosocomial pathogen, so it is very popular from human hospitals. And resistance in MRSA as well as in methicillin-resistant NAS is mediated by a MEC-A or MEC-C gene. And this gene is encoding a modified penicillin binding protein. And the bacteria who have this protein, they are resistant to beta-lactam antibiotics. And this is a problem for veterinary practitioners because most antibiotics that are approved for mastitis therapy are beta-lactams. And another important point is that resistance genes may be transferred between Staphylococci. So as you probably know, NAS are considered pathogens of minor importance for other health compared to Staph aureus, but they can transfer their resistance genes to Staph aureus, leading to higher numbers of resistant Staph aureus isolates. And if we look at the situation in humans, especially Staph epidermidis or methicillin-resistant Staph epidermidis is an important pathogen in human hospitals associated with immunocompromised patients and surgical procedures. In dairy herds, the most common mRNA species are methicillin-resistant Staph epidermidis and methicillin-resistant Staph aureus. We conducted a field study to find out more about the occurrence of MRSA and mRNAs in dairy herds in Germany. As you see here in the map, we sampled 20 dairy farms all over Germany. And herds were selected based on previous MRSA reports. The sampling procedure. We collected nasal swab samples from calves, from pre-weaning and post-weaning calves, as well as from pre-fresh heifers. From the heifers, we also collected other cleft swabs. From dairy cows, we took about 120 quarter milk samples on each farm and bulk tank milk. And from the environment, we collected swab samples from teat liners, as you see here, but also dust samples. So in total, we had about 3,000 samples that were screened for these methicillin-resistant Staphylococci. In the lab, we performed a so-called double selective enrichment method to identify the methicillin-resistant isolates. And this was used by cefoxytin-containing media. So we did not detect methicillin-sensitive or susceptible NAS, only the resistant ones. And species identification was done using MALDI-TOF. Then we detected the MEC-A or MEC-C gene, and in isolates who carried these genes, we also typed the cassette, the gene cassette of these Staphylococci. And it is important to mention that the method was developed for MRSA detection. So we used relatively high cefoxytin concentrations. And the experts might know that for mRNAs, lower concentrations are usually used. And for some species, oxacillin is the preferred antibiotic to screen for methicillin resistance. So therefore, the detection rates of mRNAs in this study might be slightly underestimated. The results. So here you see the detection rates of mRNAs in the different sample types. The highest proportion of NAS were detected in the nasal swabs of the pre-weaning calves, about 30%. And then in the post-weaning calves and heifers, the prevalence was decreasing. And at the quarter milk samples from the cows, we had about 10% positive mRNAs. And here in dark blue, you see the MRSA or MRSA prevalence in comparison. And as you see, the MR-NAS prevalence is usually higher compared to MRSA. And this is also known from other studies that did not investigate methicillin resistance. So NAS are usually or are a group of several different species and are therefore more prevalent than Staph aureus or MRSA as a single species. We also detected MR-NAS in the environment, for example, in teacups, in dust from dairy barns, and also in swab samples from automatic care feeder. Somatic cell count measurements. As you see here, the geometric mean somatic cell count in all quarters was about 114,000. In the MR-NAS infected quarters or affected quarters, we had about 158,000 cells. This was significantly higher. We also performed regression analysis, and the quarters that carried MR-NAS were about 1.8 times more likely to have a high somatic cell count. And again, here you see the well-known pattern that the MRSA cell count is much higher. So MRSA or Staph aureus is considered a pathogen with major impact on other health compared to the NAS. Here you see the cefoxitin-resistant NAS species that we detected. As you see here in light blue, we also had some species that did not carry the McA gene. So the most common MR-NAS species that did carry the McA gene were Staphylococci, lentus, floreti, epidermidis, and hemolyticus. And we detected many Staph coni isolates, which is actually very interesting, that grew on these high cefoxitin concentrations but did not carry the McA gene. The most frequently detected MR-NAS species in the different sample types. So in calves, we mainly detected Staphylococci and lentus. In heifers, Staph coni, scurri, and lentus. And in cows, mainly Staph coni, Staphylococci, and floreti. So this is also, I would like to mention this again. So Staph coni is usually considered a more environmental NAS species from previous studies. But in our study, these coni isolates were almost exclusively isolated from quarter milk samples of dairy cows. So this species seems to be more udder adapted in our study. And in the environment, we also detected scurri and lentus, as in the calves, for example. The MR-NAS species in quarter milk samples from the different farms. As you see here, we have some farms where we have almost only Staph coni isolates, and here we have a farm where we detected high numbers of Staphylococci isolates. So on these farms, the pattern of the NAS species or the distribution within the farm is pretty much cow adapted. So there seems to be one species that is on the udder of all cows. While on other farms, like these ones, we have up to 5 different species, which is more like an environmental pattern. The transfer of these resistance genes, this is actually a difficult topic. So in the lab, the transfer of this gene cassette between the Staphylococci was achieved. And we had a couple of hemolyticus isolates on 6 farms that carried the same MAC type, so the same type of this gene cassette as the MRSA on these farms. So it might be concluded that this gene cassette is transferred between the species on these farms. But on the other hand, in most MR-NAS isolates, we couldn't identify a certain MAC type. But the MRSA isolates always, or pretty much always, carried the MAC type 5. So in the majority of isolates, a recent transfer of this gene cassette seems unlikely. And in a recent study, it was also suggested that this McA gene may also be transferred independent of this gene cassette. So to sum up, we have to say that this topic needs further investigation. My take-home message. So we detected methicillin-resistant NAS in different sample types from dairy cows, young stock, and the environment. Resistance genes may be transferred to Staph aureus, and therefore we should monitor the NAS prevalence. The most common species were these phenotypic cefoxytin-resistant coney isolates, but also Staphylococcus, lentus, floreti, epidermidis, and hemolyticus, which carried the McA gene. And again, I have to mention that the study has some limitations. So the method was developed for MRSA detection, and some mRNAs isolates might have gone undetected using this method. With that, I want to thank all the farmers and the people who were involved in the study, and I want to thank you for your attention.